All right, welcome to part two of the light reactions lecture. This is going to focus more on what happens in the actual thylakoids of the chloroplast, where the photosystems are located. So when we talk about photosystems and photosynthesis, there are two different photosystems in the thylakoid membrane. Okay, and what a photosystem is it's a collection of chlorophyll molecules and other pigment molecules, not just chlorophyll, but other pigment molecules as well, that are embedded within proteins in the membrane of the thylakoid. And these photosystems act as light gathering molecules. So they light strikes them and they absorb that light energy. There are two photosystems. There's photosystem 2, which has it at its center a chlorophyll A molecule, a special chlorophyll A molecule called the reaction center chlorophyll, that absorbs the wavelength of light of 680 nanometers, which is a red wavelength of light, the best. And because of that, photosystem 2 is also sometimes referred to as P680. The other photosystem is photosystem 1. Photosystem 1 also has a chlorophyll A molecule in its reaction center that absorbs the 700 nanometer wavelength of light the best. And that is far red light, so a little beyond just plain old red. And because of this, photosystem 1 is also often referred to as P700. And actually, that can actually be a little bit better term for it because photosystem 2 and photosystem 1 can be kind of confusing terms, and I'll explain why in just a minute here. So the way that these photosystems work, we've got this cluster of pigment molecules here embedded within a protein within this photosystem. A photon of light will strike one of these pigment molecules, exciting its electrons and bring them to a higher energy level. And then that energy is passed from one pigment molecule to the next. These are referred to as antenna pigments until that energy is transferred to the reaction center chlorophyll, where it has two of its own electrons that get excited. But rather than pass their energy on to another pigment molecule, those two high energy electrons are picked up by a special molecule called the primary electron acceptor. The primary electron acceptor is a protein. It's most likely an iron sulfur protein, which means it has iron and sulfur bound to it. So our reaction center contains this reaction center chlorophyll molecule, as well as the primary electron acceptor within this photosystem. Okay, so there's our reaction center. So what I want to point out here, this is our electron transport chain of photosynthesis. So this is, we're going to have electrons being transported through these proteins embedded within this membrane. Very similar concept to what happens in cellular respiration, right? One thing I want to point out, this is photosystem 2, this is photosystem 1. So they are not named for the order in which the events occur during the light reactions. They are actually named in the order in which they were discovered. So photosystem 1 was discovered first, photosystem 2 was discovered second. The naming hasn't been reorganized. It's just left that way, probably mostly just to confuse you, high school students who are learning about photosynthesis. So that's why I said sometimes calling photosystem 2 P680 and photosystem 1 P700 actually works a little better for some people. But you do need to be familiar with both terms for these different photosystems. All right. So let's look in detail at what happens here at these photosystems, right? So sunlight is going to strike photosystem 2, one of these antenna pigments, and then that energy is going to get passed along from one antenna pigment to the next till it reaches that reaction center chlorophyll, whose electrons are excited to a higher energy level and passed off to that primary electron acceptor. Okay. Now, one thing that has to happen here, once those electrons are passed off to the primary electron acceptor, this reaction center chlorophyll has what's called an electron hole, right? It is missing two electrons that have been passed off to another molecule, and those have to be replaced. And so there is an enzyme that actually splits a water molecule into two electrons, two protons, and one oxygen atom. Okay. And those electrons then replace the electrons that were lost by this reaction center chlorophyll and pass to the primary electron acceptor, while the hydrogen ions is combine with the oxygen atom and the oxygen from another water molecule that gets split almost immediately after to form. Um, I'm sorry. That is not happening. So the water molecule gets split. The electrons are absorbed by this P6, passed on to this P680. The oxygen atom here combines with another oxygen atom from another water molecule and is released from the plant. That's where the oxygen gas comes from. The protons, the H plus ions, remain within the thylakoid. Okay, they remain within the thylakoid. So water is going to get split into an oxygen atom, and the oxygen atom will combine with oxygen from another water molecule and be released as oxygen gas. The hydrogen atoms from water are split into 2 electrons and 2 protons. The 2 electrons replace the electrons that were lost to the primary electron acceptor. The 2 protons remain within the thylakoid. So they are starting to contribute to a proton gradient, a proton motive force within the thylakoid. Okay. So now, what happens to these electrons that have been passed to this primary electron acceptor? These high energy electrons that have absorbed that light energy are going to get passed down an electron transport chain from one protein to another within the thylakoid membrane. And as they are passed down that electron transport chain, they release little bits of energy that are used to pump protons inside the thylakoid. Sounds kind of familiar, I hope. So we're building up this proton gradient inside of the thylakoid. Okay. Now, what are we going to build up that proton gradient for? Well, that proton gradient is going to be used to drive the chemiosmosis, which leads to the creation of ATP by ATP synthase, as those protons in the thylakoid will diffuse out into the surrounding stroma through ATP synthase, powering that ATP synthase so that it can phosphorylate ADP to make ATP. Okay. Now, that ATP that's made is going to head over to the Calvin cycle to be used to build carbohydrates. Remember, photosynthesis is an endergonic process. It uses energy, right? So the ATP that's made in the light reactions is going to get used in the Calvin cycle. Now, what happens at photosystem 1, right? Sunlight strikes photosystem 1, and that energy gets passed from one antenna pigment molecule to the next until it reaches that P700 reaction center chlorophyll, where 2 electrons are excited and passed to another primary electron acceptor, another protein molecule that captures those electrons in a high energy state. Now, P700 is missing 2 electrons. Where do the replacements come from? The electrons that are passed down that first electron transport chain eventually are passed to P700 to replace the electrons that it loses to its primary electron acceptor. Okay. Now, where do these electrons from photosystem 1 end up? They get passed down another electron transport chain, but they are not used. The energy in them is not used to pump protons into the thylakoid space. They are passed at a high energy level to an electron carrier that is similar to NAD, NAD plus, and that is NADP plus. And an enzyme called NADP plus reductase passes those electrons to NADP plus, generating NADPH. Okay, and this name should make sense because what's happening is NADP plus is being reduced. Okay, and that NADPH is an electron carrier that is going to head also to the Calvin cycle. Okay, so here, just kind of to show you within the thylakoid, right? Sunlight hits photosystem 2, that excites 2 electrons that are going to be passed down an electron transport chain. We have to replace those electrons that are lost. Water is split. The oxygen atom from that water combines with another oxygen atom from another water to form oxygen gas, O2, that leaves through the stoma of the leaf out into the atmosphere. The H pluses remain within the thylakoid that are formed from the splitting of that water, and the 2 electrons from those 2 hydrogen atoms are passed to photosystem 2 to replace the electrons that photosystem 2 loses. Those electrons that were excited from photosystem 2 and passed down an electron transport chain. The energy that is released as they move is used to pump more protons into this thylakoid space, generating this proton gradient, the proton motive force. What happens with those protons inside of the thylakoid? They diffuse down their concentration gradient through ATP synthase, powering ATP synthase to make ATP, which then heads to the Calvin cycle. At the same time that light is striking photosystem 2, remember it's striking photosystem 1. It's not like beams of light can hit photosystem 2 only in this leaf and miss photosystem 1. Light's hitting both at the same time, right? So photosystem 1's electrons in its reaction center chlorophyll, its P700 chlorophyll, are getting excited, and they are passed to an primary electron acceptor that then passes them to an electron transport chain where they are eventually passed to NADPH, and the ATP and NADPH made here head to the Calvin cycle. Okay, so the electron transport chain of photosynthesis uses light energy to make ATP and NADPH, which are going to be used in the Calvin cycle. So photosystem 2 absorbs light. The excited electrons pass from the reaction center chlorophyll to the primary electron acceptor. The electron in that reaction center chlorophyll is replaced by an enzyme that splits water and extracts 2 electrons from the 2 hydrogen atoms and supplies them to the chlorophyll, leaving the protons from those 2 hydrogen atoms, the H pluses, in the thylakoid, and releasing the oxygen out to the atmosphere. So water is split, the oxygen combines with another oxygen to form O2 and gets released to the atmosphere, and we breathe easier. Okay, so one experiment that was done in the early 1900s was done to figure out, okay, where does the oxygen gas that's released come from in photosynthesis? Does it come from the carbon dioxide or does it come from the water? And so a radioactive tracer, a radioactive isotope of oxygen, oxygen 18, has 2 more neutrons than the more common form of oxygen, which is oxygen 16, and it is radioactive. It will decay because it is unstable. And so what was done was water was used that had this heavy isotope of oxygen rather than just normal, the more abundant form of oxygen. And then it was traced to see, okay, which product molecule does that radioactive isotope of oxygen end up in? Does it end up in the glucose that's made, or does it end up in the oxygen gas that is released? And it ended up in the oxygen gas that was released, which was evidence that the oxygen that we breathe comes from the splitting of water during the light reactions of photosynthesis. A second experiment was done that kind of helped to verify this, and that was using the same radioactive isotope of oxygen, only having it present in the carbon dioxide rather than the water. And when that experiment was done, the radioactive isotope of oxygen ended up in the glucose that is produced as a result of photosynthesis. Okay, so this proved that the oxygen that we breathe comes from water and not carbon dioxide, and that plants actually split water. Okay, and this experiment was carried out by a biologist from Stanford, C.B. Van Niel. And at the time he was doing his experiment, people just thought, okay, the carbon dioxide is split into a carbon atom and the oxygen molecule that is released. And then the carbon combines with the water in photosynthesis to make carbohydrates, which have the empirical formula of CH2O. But what C.B. Van Niel noticed is he was working with special bacteria called purple sulfur bacteria. They use hydrogen sulfide gas instead of water in photosynthesis. And what he noted was that they produced sulfur and carbohydrates. So he was reasoning that the electrons that go to are used in the light reactions come from this hydrogen sulfide being split, and this sulfur that is released would be very similar to what happens in organisms that use water. So his reasoning was carbon dioxide plus some kind of a hydrogen source here, which could use hydrogen sulfide, water, is going to result in the formation of carbohydrates plus the release of whatever is attached to those hydrogen atoms. And sure enough, using radioactive isotopes of oxygen with plants, that's what he verified. That's what he verified, like we saw in the previous slide. Okay. Now, What we've been looking at is referred to as non-cyclic photophosphorylation, right? It was the energy from the sun, the energy of light is used to drive the phosphorylation of ADP to make ATP. And this is non-cyclic. So we got the electrons from photosystem 2 are elevated to a higher energy level, pass to photosystem 1, elevated to a higher energy level, pass to NADPH, right? Okay, so photosystem 2 generated energy as ATP, photosystem 1 generated NADPH. Okay, however, and let me go back there just a second. You can pause this if you need to. However, there's also something that occurs in many plants called cyclic photophosphorylation, where rather than passing the electrons from photosystem 1 on to NADP, they cycle back to the original electron transport chain to make more ATP, but no NADPH. Okay. And so that's why this is referred to as cyclic, because those electrons pass down their electron transport chain, get excited, and go back to that same electron transport chain. So this coordinates the light reactions to the Calvin cycle because the Calvin cycle uses more ATP than it does NADPH. So the plant needs to generate more ATP than it does NADPH. It's also thought that this plays a photoprotective role in allowing the plant to absorb more light energy without being damaged as well. Okay. And so, yeah, to make one glucose requires the production of 18 ATP and 12 NADPH. So in non-cyclic photophosphorylation, right, the electrons in photosystem 2 are excited, pass to a primary electron acceptor, move down the electron transport chain, generate a proton gradient that's used to make ATP, reach photosystem 1, get excited again, pass to a primary electron acceptor, move down the electron transport chain to NADPH. So this is non-cyclic, right? This is linear. Okay. However, in cyclic photophosphorylation, what happens is that the electrons reach photosystem 1, they get excited, they get passed to ferredoxin, which is a protein in an electron transport chain that is able to then pass those electrons back to the original electron transport chain between the first photosystem 2 and photosystem 1. Those electrons get passed down, more protons get pumped into the thylakoid space, and more ATP is generated. Okay, so brief summary of photosynthesis. Where does the energy come from? The sun. Where did the electrons come from? Water, right? Water being split. Where did the water come from? Got absorbed in through the roots. Where did the oxygen come from? It came from the splitting of water. Where did the oxygen go? It diffused out of the thylakoid, out of the chloroplast, out through the stoma of the leaf to the atmosphere. Where did the protons, H pluses, come from? They came from the splitting of water, as well as they were present in the stroma and then pumped into the thylakoid. Where did the ATP come from? It comes from the diffusion of those protons through ATP synthase, right? Which allows ATP synthase to make ATP. What will the ATP be used for? It'll be used in the Calvin cycle to start making organic molecules within the plant. Where did the NADPH come from? It came from the second electron transport chain. The electrons from photosystem 1 were excited, passed down the transport chain, and eventually transferred to NADP plus, generating NADPH. And the NADPH will be used as reducing power in the Calvin cycle to reduce carbon dioxide into the organic molecules like glucose that the plant needs to build. So Calvin cycle's coming next. That's what you're going to be watching a video on after our next class. So have a great day, everybody. If you have questions, please sign up for an academic support time, and I'll be happy to help you out. Bye.